Good afternoon. This is Loren Cordain. I'm unfortunately not able to be in the audience today and be speaking to you directly. So that's the reason for this audio presentation along with the visual. So my presentation today is going to be on dietary salt, an evolutionary perspective examining cancer, immune function, cardiovascular disease, and health. My co-author on this paper is Tony Sebastian, Professor Emeritus from the University of California at San Francisco. He worked with me on some of these sodium values that I'm going to be presenting today. First slide. I dedicate this presentation to my friend and scientific colleague, Stefan Lindeberg, MD, PhD, who recently passed. Here's a couple of cool photos of Stefan, one in 1990 performing general healthcare exams of Kitavan Islanders. This is what one study is very well known for. And this is Stefan enjoying his music at home. Next slide. If we look at the contemporary paleo diet, it's a lifetime program of healthy eating comprised of fresh veggies, fruits, nuts, seeds, healthful oils, fish, seafood, and grass-produced meats. This concept was originally brought to the forefront by my colleague Boyd Eaton in 1985 in his now classic paper in the New England Journal of Medicine called Paleolithic Nutrition. And he suggested that the food we ought to eat should be consistent with our species' evolutionary dietary background. Amazingly, this hypothesis was only first tested and confirmed experimentally 22 years later by Stefan and colleagues in 2007. So it took quite a while before it was experimentally confirmed. Since 2007, in the last decade, at least 25 or more experimental and epidemiologic studies have examined it and have demonstrated the therapeutic potential of contemporary paleo diets. And here's those studies. It's not comprehensive. There's a few that I've missed, but I want to point out that many of my friends and colleagues are in the audience today. Tommy Johnson was involved with Stefan on many of these early studies. Linda Frassetto, I know, is in the audience as well, and her colleague Tony Sebastian is my co-author on this presentation. She was involved with some of the early studies. You can see here's Tommy once again, and also Fritz Musquiat has been involved in at least one study that was contributed to our knowledge. And here's again some more of Tommy's study, Tony's, and Linda's studies. And I'd like to point out to the young lions in the audience that Pedro Bastos and Milan Fontes also have been involved in experimentally testing the paleo diet concept. Excuse me. If we look at historical studies of hunter-gatherers, it was demonstrated that salt was rarely or never added to foods. And these are some of the classic studies by Stefansson, Denton, and so forth. If you haven't read these, you may want to. Linda Frassetto and Tony have also published a good paper on this. And a colleague that many of you may have never heard of is Birger Jansen, and he did some of the very early stuff, and I happen to have privilege to his unpublished book, and there's a chapter in there which outlines the fact that unacculturated populations didn't eat salt. Well, why didn't they eat salt? Because it was generally unavailable to inland living populations. And if you look at the sodium concentrations of unadulterated non-salted foods, they tend to be quite low, whereas potassium concentrations are quite high. Thus, it makes high salt, low potassium intakes virtually impossible or implausible without added salt. Okay, so let's show you this data. This is data that Tony Sebastian and I compiled months ago, and it contrasts the sodium and potassium concentrations in a variety of foods, and then we calculated the potassium to sodium ratio in both milligrams per milligrams and also millimoles per millimolar. And you can see here that in wild and domesticated meats, sodium is much lower than potassium, yielding ratios that are much greater than 1. We find a similar situation with poultry, a similar situation with organ meats. There's only one exception here, and that's with kidney. Kidney tends to filter blood and tends to concentrate salt, so that's why you see a lower ratio here. But overall, in organ meats, way more potassium than sodium. Freshwater fish, we also see that the same situation occurring with very low sodium, high potassium, yielding a high potassium to sodium ratio. Same way with marine fish. You may think that marine fish, because they live in salt water, may have more salt, but they don't, and you can look at the overall data here. Marine mammals, reptiles, sea lions, seals, walruses, whales, and so forth. Once again, we see the very same situation with much more potassium than sodium. Eggs are a little bit lower here, but they still have more, on average, more potassium than sodium. Wild fish roe or eggs, also much more potassium than sodium. You can see here milk, even though it's not a paleo food, also has more sodium or more potassium than sodium. Whole grains, again, a non-paleo food, much higher potassium, very, very low sodium. Nuts, a similar situation. And if we look at fruits, way higher potassium, way, way lower sodium. So all fruits are a very, very good source of potassium, low sodium. Vegetables, the same situation, a little bit slightly higher sodium, but way higher potassium, yielding a high potassium to sodium ratio. Non-starchy root vegetables, again, the same situation, much higher potassium, lower sodium. Starchy root vegetables, again, the same situation. Legumes, although not necessarily a contemporary paleo food, we see the high potassium, low sodium, and these are the ratios. Dried fruits, again, same situation. Here's miscellaneous foods. Some people may like to drink a little bit of wine or beer on a paleo diet, and you can see these are, in terms of potassium to sodium ratios, healthful. Marine seaweed, we also see a higher potassium and sodium ratio, although there are certain exceptions to this. Coconut water, also the same situation. This is my colleague Jenny Brand Miller's data, and it comes from 759 wild Australian hunter-gatherer plant foods. And you can see here, this is an enormous database, once again showing way more potassium than sodium, a very high potassium to sodium ratio. This is Jenny's data this is her unpublished data for wild Australian animal foods. And you can see this includes such things as insects and organ meats and birds and reptiles and fish and marine mollusks and so forth. And once again, way more potassium than sodium, yielding these kinds of average ratios. There is one exception to the rule here when we look at unsalted food, wild foods, and this is marine shellfish. And if you examine this table, you can see that about half of the marine shellfish tend to have very low potassium to sodium ratios, whereas about half of them have a little bit greater than 1. And the reason for this is that shellfish, marine shellfish in particular, are osmoregulators, and they tend to maintain the osmolality of their environment. However, to date, it's not known if the high salt in shellfish results from um, sodium chloride or potassium chloride or both. So that data needs to be resolved. However, you may think that if you theoretically ate an all marine shellfish diet without any plant foods, then you could get an overall dietary potassium to sodium ratio below 1. However, that's really an impossible physiologic situation simply because shellfish are very, very high in protein, low in fat, low in carbohydrate. So to eat more than 40% of your energy from protein becomes toxic. So once again, a high salt diet would be even impossible with these foods. So let's look at our conclusions here. The daily caloric intake for men ranges between 2,400 and 3,000 kilocalories. For women, 2,000 to 2,400 kilocalories. And you can vary the plant to animal subsistence ratio any way you want. You can go all the way up to 65% plant food and 35% animal food, or all the way down to the opposite, 35% plant food, 65% animal food. And using those ranges, which we have actually calculated for other studies, it's virtually impossible to eat more than 2,400 milligrams of salt per day for adult men and women. And moreover, more importantly, if you eat unsalted foods, it's impossible to eat a potassium to sodium ratio of less than 5, and more likely your sodium or your potassium to sodium ratio will be between 5 and 10. Now let's contrast that to the average US diet, which contains more sodium than potassium, and it yields a potassium to sodium ratio of 0.77. Again, this would be impossible under any normal situation eating normal unsalted foods. So let's look at the sodium and potassium concentration of sandwich ingredients: bread, processed meats, cheeses, and condiments. These are favorite foods both for Europeans and Americans. And you can see here that the sodium potassium concentration of processed meats are way high, way more sodium, way lower potassium, yielding a ratio of much less than 1 for the potassium to sodium ratio. And contrast these numbers to fresh unsalted meats, and you can see that fresh pork, lamb, and beef has very low salt and very high potassium, yielding this kind of a ratio. Cheeses are in a similar boat to processed meats. All cheeses are made with added salt, and here's the average value for a variety of favorite cheeses. So much higher sodium, much lower potassium, whereas yielding this ratio of way less than 1. And milk, which is not, salt is not used in the manufacture of milk. You can see then that unadulterated milk has more potassium than sodium as compared to cheese. Breads are in the same situation. Breads are one of the highest sources of salt in the Western diet, and you can see here that breads, because they use salt in their manufacture, contain very high sodium, very low potassium, yielding this ratio. Whereas if you contrast that to unadulterated whole grains, which I'm not necessarily saying are contemporary paleo foods, they have very, very low salt and high potassium. It goes without saying that sandwich condiments contain enormous amounts of sodium, and you can look over here. This is the sodium milligrams per 1,000 kcals. The average value is just completely out of control, and potassium quite low, and a very low, less than 1 potassium to sodium ratio. So whether it's table salt, sea salt, or processed foods, there are well-recognized diseases from a high salt intake: hypertension, stroke, osteoporosis, kidney stones, stomach cancer. More recently, high salt diets have been shown to promote chronic inflammation, dysregulate the innate and adaptive immune systems, promote and initiate cancer, underlie autoimmune disease, and promote cardiovascular disease via disruption of the endothelial glycocalyx. More obscure diseases are ear ringing, which is known as Meniere's syndrome, insomnia, motion sickness, asthma, and exercise-induced asthma. What I'd like to focus on this presentation is the more recent observation showing that high salt diets promote and initiate cancer. They tend to underlie chronic inflammation, which is responsible not only for cancer, but it's also responsible for cardiovascular disease and also autoimmune diseases. So high salt diets dysregulate the innate and adaptive immune systems, and that underlies chronic inflammation and all of these other diseases that we've shown. Okay, so let's look at how a high salt, low potassium diet tends to promote cancer. My colleague, as I mentioned earlier, Birger Jansen, who is no longer with us, he died in 1997, has shown that many common cancers are promoted and initiated by high salt and low potassium intakes. He was one of the first guys that got a handle on this, and he was a very brilliant epidemiologist, and you can read his literature that was published in high quality journals back in the era. And Jansen's observations were confirmed by Jacobs and Pienta in 1991, who exhaustively reviewed human, animal, and tissue studies. And this is what their comments were. Generally, a high potassium to sodium ratio is associated with decreased cancer incidence. Conversely, a higher sodium to potassium ratio with a higher absolute sodium concentration associated with higher risk of cancer. So this is some fascinating earlier work that underlies the idea that high salt, low potassium promotes and initiates cancer. Well, how does a high salt diet seem to do this? There's some really recent information that's only been out for the last 5 years or so, which reverses the dogma of 50 years, suggesting that high dietary salt is increased when high dietary sodium is increased from low to high levels. Total body sodium and total body water increase until daily sodium excretion again equals intake. This was the dogma of 50 years of prior physiological works. Balance studies using isotopically labeled sodium, 23 sodium, in conjunction with MRI probes have shown this not to be the case, and that large amounts of sodium are accumulated in some tissues without commensurate increases in water. And so this work comes out of Tzitzik's group's data from the last 5 to 7 years or so. So accordingly, many tissues, erythrocytes, skin, muscle, kidney, and brain, act as sodium reservoirs in which they contain sodium concentrations much greater than plasma concentrations. The first study to examine a high salt intake in conjunction with 23 isotopically labeled sodium and MRI probes was done by Resnick and colleagues back in 1994, and they were interested in seeing how hypertensive subjects responded to salt loading or a high salt diet, 200 milliequivalents per day. And they examined the intracellular sodium concentrations, and they found out that both normal and hypertensive subjects increased the intracellular erythrocyte or red blood cell sodium concentrations. This 23 NA-MRI evidence has now gone over and looked at salt how salt loading influences other tissues, skin, muscle, kidney, and what they found is that these tissues then contain greater concentrations of sodium than what is found in plasma. The normal concentration in plasma is about 135 to 145 millimoles per liter. So one of the interesting outcomes of all of this evidence is that the MRI data shows that salt loading produces these reservoirs of high salt that in various tissues that is not simply benign, but is integral to chronic inflammation. So salt loading increases total body salt via these reservoirs, and now there's a plethora of evidence to show that it produces inflammation through a variety of markers, and you can see these citations here, and you can examine them if you would like. So this is just further data showing that chronic inflammation is produced by increases in total body sodium from a high salt, low potassium diet. Now, let's take a look at cancer promotion and initiation. Cancer maintains 5 developmental stages: initiation, promotion, malignant conversion, invasion, and metastasis. In virtually every stage of cancer, inflammation plays a crucial role. And here are the citations indicating that we see inflammations at all stages of cancer. So high salt, low potassium diet increases total body sodium, which causes inflammation, which acts at every level of cancer development. Well, how does this work? At every stage of cancer, how does an elevated intracellular salt initiate and promote cancer? Well, one of the first things it does is it causes DNA damage via changes in osmolality of the cell. DNA damage causes chronic inflammation. Elevated intracellular salt induces reactive oxygen or ROS species, which also promote inflammation. High salt diets promote synthesis of vascular endothelial growth factor. So this is a cytokine which is associated with angiogenesis, the formation of new blood cells, which all cancers require. Salt also adversely influences the innate immune system, particularly the way it changes the balance between M1 and M2 macrophages. High salt diets promote a specific type of metabolism that only cancer cells exhibit, which is called Warburg metabolism. And high salt diets cause T cell activation or adverse changes in the adaptive immune system, which tend to also promote cancer. So elevated intracellular salt causes DNA damage. It's been shown in vivo and in tissue studies that elevations of osmolality from 300 milliosmoles per kilogram per water to 500 to 600 by adding salt causes DNA strand breaks. And this data was been shown primarily by Morris Berg and Dimitrieva. Goes all the way back to the mid 90s, early 90s, or not 90s, early 2000s, mid 2000s. DNA breakage and repair promotes inflammation, and inflammation we know is intimately related to the development of cancer. These are the references showing that effect. In cancer, elevated intracellular salt promotes the cancer process via production of reactive oxygen species, ROS are superoxide hydrogen peroxide hydroxyl radicals, and these promote DNA strand breaks, particularly during periods of environmental stress like excessive UV radiation, pollutants, and high salt diets. Reactive oxygen species produce inflammation, and excessive ROS may lead to cancer, shown by a number of experiments. And elevation of salt in tissue cultures and in vivo studies increases ROS production. In cancer, we also see an elevation of vascular endothelial growth factor, VEGF, and we know that salt tends to promote that. There's a process called angiogenesis, which is the creation of new blood vessels that are absolutely critical to tumor growth and metastasis. And so the induction of VEGF plays a central role in activating pathways, NFAT5, AKTP, and PI3K, which tend to cause angiogenesis. It's been shown in randomized controlled trial that high salt diets in both humans and animals increase circulating concentrations of VEGF. In cancer, salt acts to promote the process by activating macrophages. Macrophages are part of the innate immune system that engulf and digest cellular debris, microbes, and cancer cells. There are two specific types of macrophages we're interested in. M1 macrophages that encourage inflammation and promote cancer, whereas M2 macrophages decrease inflammation and promote tissue repair. High salt diets activate M1 macrophages while simultaneously inducing an M2 environment with poor phagocytic ability to engulf and remove cancer cells. So you can see here that when you go from a low dietary salt to a high potassium environment, it increases total body sodium in a variety of tissues. Finally, in cancer cells exhibit a specific type of metabolism called Warburg metabolism, in which they have an inefficient metabolism of glucose and produce large amount of lactate and little ATP. First identified by Otto Warburg in 1924, all cancer cells tend to exhibit this Warburg metabolism. And it's known that high salt diets exaggerate Warburg metabolism, particularly in breast cancer cells, by increasing osmotic stress. So remember that sodium changes intracellularly, will change the osmolality intracellularly, and this is how it's thought to elevate Warburg metabolism. High salt diets promote cancer by activating T cells. T cells normally come in their naive state, which is called CD4 plus T helper cells, and they form an essential part of an antigen-specific adaptive immune system. And these cells function is primarily governed by the microenvironmental cues that they encounter locally, particularly at tumor sites. So local tumor inflammatory conditions cause naive T cells to be recruited to the site, and a high salt diet induces these naive T cells to differentiate into Th17 or T helper cell 17s, and the Th17 cells increase secretion of the pro-inflammatory cytokine IL-17, which are released into the tumor microenvironment and seem to induce angiogenesis by causing an increased secretion of VEGF, and this process then leads to enhanced tumorigenicity and cancer metastasis. So if we look at a high salt diet, it not only causes this pro-inflammatory effect on naive T cells and the formation of Th17 cells, but it also impairs T regulatory cells. These are also key elements of the adaptive immune systems. Tregs normally release immunosuppressive IL-10 cytokine, and high salt diets promote interferon release from Tregs and abolish their normal anti-inflammatory and immunosuppressive effects. So taken together, the suppressive effects of high salt diets on Tregs support Berger-Janssen's epidemiologic evidence that shows that a high salt, low potassium environment initiates and promotes all types of cancers. So to date, the focus of this presentation has been on the more recent observations about high salt diets and adverse effects on health and well-being. Cancer promotes and high salt diets promote cancer and their initiation via chronic inflammation and via dysregulation of the innate and adaptive immune system. Let's also take a look now at how these changes may influence the development of autoimmune disease, and then finally we'll look at how cardiovascular disease may be influenced by how high salt diets influencing and disrupting the endothelial glycocalyx. When the immune system cannot distinguish self-tissue from non-self tissue, it mounts an immunological attack on self-tissues. This is what autoimmune disease is. And though the etiology of autoimmune disease is incompletely understood, a number of recent experiments show that a high salt diet likely underlies the development of autoimmunity. So as I mentioned earlier, high salt diets activate Th17 cells via this pathway, the SGK1 pathway, to produce the IL-17 cytokine, whose presence is associated with inflammation and numerous autoimmune diseases, including rheumatoid arthritis, lupus, multiple sclerosis, Crohn's disease, and ulcerative colitis. Unfortunately, no randomized controlled trials have been conducted in humans testing low salt diets in autoimmune disease, but I suspect that we'll be seeing some of those shortly. Well, it's been known for years that high salt, low potassium intake is associated with cardiovascular disease, and the mechanisms underlying this are likely endothelial cell inflammation and other inflammatory processes. This is some interesting data here. This is data that was published in Circulation a few years back by Cook and colleagues, and this is the trials of hypertension prevention, or TOF studies, that showed that there was a linear decline in cardiovascular risk cardiovascular risk and mortality with decreasing sodium intake. This actually is the all-cause mortality over 20 years in TOF. And you can see that the relationship remains linear almost down to the lowest levels of salt intake and all the way up to the highest values. So some people have criticized this TOF data, suggesting that there actually is a nonlinear relationship, a J-shaped relationship or nonlinear relationship between salt and all-cause mortality. So they suggest that at low salt intakes, there's actually an increase in mortality and vice versa. The problem with that interpretation is that they were based on erroneous 24-hour urinary sodium data. And so recently in this group's study here, Lacherl et al., 2015. It's been shown that 24-hour salt ingestion and sodium excretion in a controlled environment are a total joke. There's absolutely little or no relationship between 24-hour urinary sodium and salt ingestion. More well-controlled studies in controlled environments have shown that it takes 3, 4, or more measurements of 24-hour urinary excretion to get at anywhere near the amount of salt that is ingested. So the claims that cardiovascular disease has a J or a nonlinear relationship are probably a result of these erroneous urinary data that has been presented. Let's take a look at how a high salt diet disrupts the endothelial glycocalyx. Here's a classical visualization of an artery cross-section until about 1994. And notice that the inside of the artery is shown to be endothelial cells rather than the endothelial glycocalyx, this kind of hairy structure that actually lines the inside of arteries and blood vessels. The endothelial glycocalyx was first detected in 1966 using electron microscopy with special staining techniques. And the function of the glycocalyx was always speculative until 1996, about 20 years ago, when Vink and Dooling concluded that the glycocalyx may represent the true active interface between blood and the capillary wall. So indeed, that's what its function is. And the glycocalyx is a real dynamic structure. It's shed in response to inflammation and continually resynthesized. And the shedding of the glycocalyx is a necessary process in response to injury or infection. It allows leukocytes, white blood cells, and platelets to infiltrate the endothelium, the inner layer here, and enter the intima. The intima is the area below the endothelial cells, and those leukocytes then can begin healing and disposing of diseased tissue underlying the endothelium. So under normal conditions, when the glycocalyx is not shed, then it prevents these blood clotting elements like leukocytes and platelets from entering into the intima where they can cause atherosclerosis. The glycocalyx also regulates the entry of LDL cholesterol into the intima, and the glycocalyx transmits flux of either turbulent or non-turbulent flow to the endothelial cells. And if you look at where atherosclerosis occurs, it tends not to occur in areas where we have laminar or smooth flow, but it occurs almost exclusively in areas where there is turbulent flow, like in branching of arteries and so forth. So this is what this little diagram shows right here. So that shows then that an intact glycocalyx before it's shed acts as a real gatekeeper for the immune system, leukocytes, monocytes, naive T cells, and other factors. And when it prevents these from getting into the intimal or subendothelial space, then atherosclerosis cannot proceed. So here's a schematic diagram of the atherosclerosis process without the glycocalyx. So notice then that the fuzzy hairy part lining the inside of arteries is stripped away here. So this would be equivalent to a shed glycocalyx. And when the glycocalyx is shed, then it allows T cells and monocytes to attach to adhesion molecules, and through a process called rolling and other factors, they can enter the intima, or the subendothelial space right here, And this is where all the action occurs in atherosclerosis. And you notice here that monocytes are activated to become macrophages via inflammatory mediators, and they tend to gobble up LDL cholesterol. And when they do that, they become foam cells, and the foam cells then ultimately are what cause atherosclerotic plaques. So how does salt influence this whole process? Salt causes glycocalyx shedding, and it initiates immune system entry into the intima and allows formation of foam cells along with inflammatory mediators. Normal concentrations of salt are normally maintained at 134 to 148 millimoles per liter, and high salt diets can easily elevate these numbers to values as high as 143 to 150 millimoles per liter. And when these values of salt get to this level, it causes endothelial cell stiffness. It causes endothelial cells to reduce their synthesis of nitric oxide, which normally suppresses inflammation and adhesion of leukocytes and platelets to the endothelium. One of the questions that came up is this a salt effect or is this a aldosterone effect? And so using aldosterone synthase knockout mice in which genetically they can't synthesize aldosterone, it's shown that sodium alone at levels of 150 millimoles per liter increase endothelial cell stiffness by 44% and suppress nitric oxide production. So this data suggests a direct salt effect and not a aldosterone effect. So let me kind of recapitulate here. We talked about earlier in the lecture that consumption of normal unsalted foods demonstrated it's virtually impossible to eat more than 2,400 milligrams of sodium per day for adult men and women. The only way that could happen would be if you were to add salt, mined salt to your food. Moreover, it shows that consumption of normal unsalted foods that show that it's virtually impossible to eat a sodium to potassium ratio of less than 5, and in most cases it's between 5 and 10 times more potassium than sodium. Contrast that to the U.S. daily diet that contains 3,500 milligrams of sodium and 2795 milligrams of potassium, yielding a ratio of 0.77. These kind of ratios are literally impossible. Values below 1 simply cannot happen if you eat normal non-salted foods. So the average US and probably average European diets are right around this value. So what do we know about consumption of the typical Western high salt, low potassium diet? Well, people have thought for years that it might promote hypertension in some people, but not in all people. Moreover, it's important in showing that it likely promotes and initiates cancer through chronic inflammation, changes in the innate and adaptive immune system, and promotion of autoimmune disease and cardiovascular disease via disruption of the endothelial glycocalyx. So, the paleo diet, what is it? It's a naturally low salt lifetime program of eating. Do you choose this or this? Or do you choose to slice off some of this and mix it with this and call it a sandwich? Do you choose this or this? Fresh vegetables or pizza? How about fresh salmon or a hamburger? Fresh fruits and vegetables or cheeses? Fresh fruits or hot dogs and potato chips? Pasture or grass-produced eggs or bacon, fresh nuts or French fries, a nice shrimp salad with avocado and tomatoes, or a sandwich. The choice is yours. Thank you very much.